With a vision to create, collaborate, and contribute to science, today let me give you a tour of how to design your saliva study. So, as usual, before I proceed, I would like to give my dedicated prayers to my father, Dr. Kamalesh Chandra Roy. I talk about him because he is the inspiration and behind whatever I do today and whatever I am today. So he was the first dentist of a small town named Berhampur in district Murshidabad in West Bengal. All right, so let's move on to the session. And as many of you have been attending my webinars earlier, so you know that this is the best way I love to describe saliva, because uh, very well told by Dr. Irwin Mandel that saliva lacks the drama of blood, the sincerity of sweat, and the emotional appeal of tears. Well, this is so rightly and aptly described, and today I will try to give you some views on how you can actually think of exploring this wonderful biological fluid more. And if anyone else has joined, hi Pooja, how are you? So that's nice. I feel it's a Friday night and people are partying. So, anyways, I will continue my discussion here, and you can write your questions even during the webinar in the comment box in the comment, and I'll try to answer them after the webinar as soon as possible. Okay, so today we are going to discuss today about how to design your saliva study. That's very important because even before we think of saliva, um, we need to understand about it, and even if we want to try to explore it, we need to understand what are the aspects that we should be considering before even we think of working with such a wonderful biological fluid. So these are some of the subheadings which I will be discussing today. So the first is definitely is to understand the analytes. I mean the things that you are actually trying to focus on analyzing. The second is very important is how to collect saliva sample. There are many considerations which I have been discussing in my previous webinars, but today I will focus on some more important aspects which you must consider even before you start writing your proposal. Um, I hope all is good. You are able to hear me and see the slides. Um, if it is so, please kindly write in the comment. Okay, and next point that I discuss is to check the latest reviews and references. And very important thing is when you are trying to outsource your work. If you have a laboratory, well and good, but if you do not have, then it's very important to choose a correct laboratory or your outsourcing partner. I will speak on this aspect. And a bit of understanding of statistics is also very important. We don't need to be a statistician, but definitely we need to understand certain areas which will give us a better understanding when we start analyzing our data, or what we can more focusedly understand why exactly we are trying to do this study, or what is our aim. That would be more clearer. I will give you certain examples and try to explain this aspect in my upcoming slides. And I know there will be many questions, but well, I will definitely, it is a very small scope in the webinar, so as much possible I will be focusing on solving the FAQs, commonly asked, frequently asked questions, particularly related to saliva storage, handling, etc. Okay, let's plunge in now. So as you know that how versatile saliva is, and what is saliva? It's definitely a biological fluid. It's a and it drains through. It comes all the way, flushing and through the salivary glands, through the many cells, the acinar cells, and so on and so forth. And it comes out in your mouth. Okay, so. Uh, why we talk so much about saliva today is because it is as important as any other biological fluid. Now, just like blood. So the best part of saliva is that there are the biomolecules which we try to analyze can pass from blood to saliva. That is how saliva becomes so very versatile. It has enzymes, it has cells, it has dissolved gases, it has inorganic substances, trace elements, proteins, immunoglobulins, DNA, RNA. I mean, the list goes on. Well, so um, to to very particularly to understand your analysis is important, because before even we try to determine, try to think what we are exactly going to determine, I will take you to the next slide and then come back here. See, uh, the thing that makes saliva versatile is because the biomolecules travel from blood to saliva through different routes, like diffusion, like active transport, ultrafiltration, etc. So when I am trying to analyze a particular component in the saliva, then I need to understand these biological mechanisms a bit, if not the whole thing, because these cell biology components will ultimately reflect on the amount of that analyte present, the concentration of the analyte, etc., and how easily I can whether it will be easy for me to determine or it will be difficult, all those aspects. So I will give you one example. Like for example, I go back to the previous slide and you see, if I am here to test, or my analyte is DNA, because maybe you might have heard about the SNPs. These are single nucleotide polymorphisms that are present. Okay, let me make it very simple. It's nothing. It's like, for example, suppose take any disease X, okay, and it has a gene. So now this gene will have different forms, okay. So one form is present in me, other form is present in you. So in that way, because say a form of that X gene is present in me, just because of a very minute difference, okay, between the DNA base pairs, and you have a different set of base pairs, that makes you to have the B form of that X gene. So in that way, what happens for the same disease is the same gene, but there are two different forms, and that makes both of us have different susceptibility to the disease. I mean, I might be more vulnerable to that disease, you might be less. So anyways, that was different thing that I was talking about. So what I mean to say, let's come back to the topic. So when I'm trying to say do some SNP analysis, so my analyte is salivary DNA. So that's again, I have to see when I do salivary DNA analysis for a particular thing. I'm trying to see whether I am doing a whole genomic analysis, or I am trying to find out any bacterial DNA. So in each of the cases, the entire design protocol changes, because if I am trying to take the bacterial DNA, or I'm trying to study the whole genome, then in either of the case, the other becomes a, I mean, what to say, other becomes not wanted, unwanted thing. Like for example, if it is the whole genome DNA, then I don't want bacterial DNA contamination, right? So that's the thing. Again, for example, if I am trying to do certain analysis of biomarkers, I mean these are proteins, and most commonly, like for example, you have heard about much about C-reactive protein, that is CRP, then cytokines. So all these are biomarkers of inflammation, right? So what happens if, for example, you know, uh, just now I told you that there are different aspects where the biomolecules can travel from one with different mechanisms from blood to saliva. But now for in this example, if I talk about CRP, now CRP originates largely from liver and it is a very big molecule. So it cannot pass into the circulation, into the salivary glands by diffusion or ultrafiltration. What happens is there is something called as GCF, that is gingival trabecular fluid. It's the fluid that flows out from the junctions between the teeth and the gums. So CRP goes and sits there. It that's a better seat for the CRP, and when saliva comes, it mixes with saliva through the JCF. So in this case, if I'm trying to do CRP, it's better to collect whole saliva. So just see that, um, and I will use passive rule method maybe. So or if it is very specific, I will use different other tools. I'll talk about it. So just by changing whether it is a biomarker of inflammation, I mean whether it is a protein component or DNA, I mean the analyte. So it becomes, the scenario becomes totally different, right from um, you know how we are trying to do, what methodologies we will use, how we will collect sample, everything changes. Okay. How to collect saliva sample? A very important thing is that passive drool has been, as you see in the picture, passive drool has been a gold standard. So saliva can be collected through passive drool as well as through swabs, which are commercial swabs now available in the market. Now, passive drool collected samples can be used to test any analyte. But when we are looking forward to some specific analytes which might be, less in amount, or which might be very, very, very specific, and we don't want other interference from other components like DNA, other mucin, all those aspects, then we might choose some specific swabs over passive rule. Also important is when passive rule is not feasible in many cases, like for example, in these cases we have to go for swab collection, and it is also very important to be aware that all swabs are not the same. Like for example, if you see in this picture, this is one swab which is used for collecting saliva sample from infants, from small babies. And when we need DNA, all right, when we are trying to analyze salivary DNA as a whole, and along with the whole saliva, we will also try to take some scrapings of the buccal mucosa, because then we get the DNA, the picture of the genomic DNA in entirety. So just you can understand that these recommendations of the collection devices um, depends on the whether you are analyzing the analytes now, whether they are in low component, or whether the research participant, the age is very important, whether it is an infant, this child, adult, any elderly, or sometimes if it is necessary, people work on animals too, and also whether the participant is capable of understanding simple instructions that you give. Like for example, if you work with people who are having mental illness, or children who are who have some special needs, uh, like for example, autistic children or children with some syndrome. So in all those cases, or maybe where you cannot expect passive rule due to some pathology in some patients. So in all those cases, different swabs are recommended for different purposes. And another important factor, just if you see here, is to consider the frequency, timing, and volume of sample that you request from the participants. Because again, the the participant has to be comfortable. Um, Plan it in a way so that they are very ready to give you the sample, and also it's convenient for them. And request always an excess of sample than required, maybe not much, but maybe somewhere around 300 to 500 microliters more. Because generally when we go for immunoassays or other assays, we really need it in microliters, uh, sometimes even less than 100 microliter, but yeah, it's always good to be on a safe side. Very important point is to check the latest references. In today's time, salivary biosciences is being expanding like anything. Uh, if you if you are not updated about what is going on, then you might land up with a study which has already been published or which is already been done. So it's always good to check the latest research updates from well-known sites like PubMed and others. This point is very important because most of the time, like for academicians and clinicians who might be interested in doing some work with saliva, they will not have the entire facility. So you need an outsourcing partner, meaning to say that you outsource your work to certain laboratory. So this is very important point because your entire time, money, energy, you know, everything, the most important, the science that you are trying to contribute to, all depends on who is your outsourcing partner. So you must select a laboratory who are expert in handling saliva samples. Though the basic procedures might be similar, but it is not as same as handling other biological samples. So make sure the lab follows proper guidelines and rules and protocols, which is very important. And also, I would suggest that there might be and there must be um, many laboratories who would be providing you services plus, but please do a comparative study about the rates they provide, um, and more importantly about the type of work they do. That is very, very important. And the analysis part is very important, whether they help you out through that also. All these things are important because it will ultimately affect your hard work at the end. Okay. Now, understanding statistics in sample analysis. I know it's a very vast thing, and in one slide, I really cannot explain all statistics. But let me just try to tell you what I mean to. I mean, what is my intention in keeping this point is that, see, some simple things. It might sound simple, like for example, you are collecting saliva sample. You have stored it in a not in a proper way, or you have somebody who has tested, or you have tested yourself, and your pipetting was wrong, or you just made some mistakes. So all these things, errors. I mean, nobody's perfect. There will be errors. So what? But as a whole, unsystematic errors creeps into your um, results finally. So so what stats these particularly these statistics helps in to analyze is what is the kind of error that has crept in. Rather, if I put it another way, how precise is your study? How perfect is your result? How reliable is your result? So that is very important. So one of the most spoken of component which I want to focus here in this webinar today is, you might have heard about variability. See, coefficient of variability. Well, if I speak like this, it sounds something very complicated. But variability is nothing but, see, as I mentioned, all these errors because of, say, wrong sample handling, wrong sample storage, wrong procedures, small errors have crept in. Like, for example, somebody was pipetting the saliva sample, and instead of in the pipette, in the tip, instead of 2 ml, sorry, instead of in 100 microliter of sample which was aspirated by the pipette, there might be a bubble. So that bubble means that's an air bubble. So if there is an air bubble, what will happen? You are not actually measuring 100 microliters. You are measuring less. So this kind of errors, which are always not intentional, always creeps in, and it affects the results. And definitely then it affects the precision of your work. So for example, um, what is this variability, or what is this coefficient of variability? There are things written over here in the slide, so don't worry, I will try to explain you in a very simple way. There are two things when we talk about coefficient of variability, particularly when we focus on immunoassays as in ELISA, that is enzyme linked immunosorbent assay. So if you have read papers, or if you have seen books, you might have come across this term, and generally the results are, when we interpret the results in immunoassays, this is one of the commonest thing that has to be considered for the precision of the result. So what is this interassay and intraassay? I'll come back to the slide. So this, what is intra? Intra means within. So intra-assay variation is that variation of results within a data set obtained from one experiment. Okay, that is what is written here. So this particular variation, if we calculate the intra-assay CV, that is intra-assay coefficient of variation, this helps us to monitor the deviation within the same assay. I will explain you what do I mean the deviation within same assay. For example, I have a saliva sample, okay, and now I have made 5 dilutions of it, and this, like for example, uh, 1 is to 10, 1 is to 20, 1 is to 30, like that. I have made 5 dilutions of the same sample, and I will analyze it. Now I have made duplicates of these dilutions. So each time when I measure the analyte in this, I will get some results. The next set, which contains the same thing, I will measure, I will get a little bit difference, because it cannot be, it won't be perfect. So this is what is meant by intra-assay variation. You are doing it at one shot of time, but it is the deviation within the same assay. So what is inter-assay deviation? Inter-assay deviation is very important, because when do we tell that a thing is precise? Okay, this test is reliable, means the number of repetitions of that result of the number of repetitions of the same experiment give you the same results and not different. So when your results are so that it doesn't actually differ much, so that means your test is reliable, right? So similarly, that's the thing which we try to understand, that suppose today I did one set of experiment, one set of data I have analyzed certain samples today. Next day I repeat the same experiment, same thing, we're doing all the same things, but I find some discrepancy. Third day I repeat, fourth day I repeat. So all these data when accumulated together and analyzed, the result that we see is expressed in terms of interassay coefficient of variation. I'll now go back to the slide and see in cases mostly, see how reliable, what kind of information you get out of this. So interesting. See, for example, interassay percentage, it is expressed in percentage. So interassay percentage CBs of less than 15 are generally acceptable. So let just note in the example that if experimental results is there with poor intra-assay, if the experimental results have coefficient of variations, intra-assay coefficient of variation less than 10%, that means there was very bad pipetting that was done on the part of the laboratory technician. That is the information that we get. So in this way, it is very important to understand a bit of uh, statistics, so that we have a better understanding or vision of what our results would be, or what we want to, what we are trying to get out of this, any of the study that we are planning. And also, when the results are obtained from your outsourcing partners or the third parties, you will be able to identify this kind of errors, even if they are not honest enough to tell you. So it's your protection for your data set. Okay, so there are some common questions which we generally face, and people ask, like, how long can saliva, for each analyte be stored before use? So it's always recommended that all saliva samples that you collect should be frozen as soon as possible after collection, and refrigerate immediately at 4 degrees centigrade after collection, and it should be transferred to -20, that is your the normal fridge deep freezer that you have in the normal refrigerator, that is -20. But yes, again, this cannot be generalized, because there are certain salivary analytes like salivary DNA, etc., where you have this they are quite stable, and even in certain cases of salivary RNA, but no, it's a big no for salivary proteins. You have to refrigerate it, because nowadays we have certain technologies which are available, like I was talking about the saliva collection tools. There are specialized tools which will have some stabilizing solutions, which will help to keep your salivary DNA or RNA stable at room temperature until it gets transferred to your laboratory. But it is not that with the salivary proteins. Okay. Next question is, sometimes people ask that, okay, can we collect samples from with cotton? No, it's absolute no. It's not recommended, because cotton is organic in nature, and it has been seen to interfere with different analytes in the saliva when you do the estimation, and this has been proved by research. Well, another question is that, why do I have to freeze and centrifuge saliva samples before testing them? Yeah, very important. See, this is particularly for whole saliva. In whole saliva, there are glycoproteins, that is mucins, we know that. So because of mucins, the saliva is thick and sticky in consistency. So using fresh saliva for quantitative estimations leads to inconsistent results due to these mucins. So once you freeze, these mucin proteins will precipitate and fall out of the solution. And once then you thaw the samples, and that means you again bring it back to the room temperature. That is what we mean by thawing. And then we centrifuge. So that will totally precipitate the mucins or other cellular debris and food particles that might have been present in the saliva sample to the bottom. And you can use the clear supernatant for your further analysis, because this will give a more accurate and consistent result. Well, another question is, how many analytes can be analyzed from one sample? Well, a single saliva sample can be used for the measurement of multiple analytes. And if you are looking for DNA, you can use the saliva sample, same saliva sample for like whole saliva, you can use for DNA analysis also. It's only that you have to understand and separate it out in different aliquots, so that you use them for your future work. For if you want to take one saliva sample and say, you have an intention to analyze many different or multiple analysis of multiple analytes, then you have to just store the sample in a better way in different aliquots, not in one single vial. Okay. And yes, another very important question is, how should I dispose of saliva samples after testing? Well, that has to be very institute specific, because saliva is considered as a class 2 biohazard, and you have to check with your organization and the policies that they followed regarding biohazardous waste. This disposal should be of the saliva sample after use should be consistent with that. Okay, and this was all from today's webinar, and I would request you all to follow our new Instagram with hashtag Croinas, where you will find interesting news about saliva research going on in the world, in the entire globe. We try to post interesting facts about saliva, the researches, the tests, the applications of saliva that is coming up and that is happening in the entire world. And also, I would request all of you to join the Indian Academy for Clinical and Dental Genetics group, where we have started a new endeavor of uh, hashtag Let's Discuss on Wednesdays. This is basically a discussion which we can take forward according to your recommendations and your wishes, like where you can ask, suggest, and share anything pertaining to your research, or you might also suggest what kind of discussions you'd like. If there is if you have a particular paper, say for example in a journal paper that you want to get discussed, you want to have a better understanding, then we can take that up, and we can just discuss on that. And I think that's a better way of sharing knowledge and understanding. Well, and that is all. And please visit our website www.genedent.com for our more products on saliva, as well as our research services and research consultancies that we provide. And thank you so much for being with me here. And I hope I'll be doing more webinars in the future. And please. Give your suggestions, your valuable feedbacks, and any other specific topic related to saliva that you want me to cover, then I can definitely think on and bring those things, those topics as in the webinars.